All right, action potentials. So, we really want to make sure that we understand action potentials because this is where we're going to kind of branch off to a lot of physiology. Action potentials are the way that our body actually communicates and sends messages through the nerves, through the muscles, through everything to allow our body to function. So, we want to make sure that we understand this super, super well to understand uh, later on, like the pathology, especially, so that we know what can go wrong in all of these situations. All right. So, an action potential, by definition, it's a rapid, transient. That means it's moving, it moves, it transfers along. It's a moving change in the electrical potential across a cell membrane, specifically in neurons and muscle cells. It propagates or it expands, it continues, it passes along the cell membrane, enabling the transmission of signals. Action potentials demonstrate what's called an all or nothing principle. That means every single action potential, no matter where it is, it's going to have the same amount level of force. It either occurs or it doesn't. Okay, it's always consistent. It has a specific range where if an action potential is triggered, it always has the same intensity and it travels down. One action potential is not more urgent or more forceful than another. They're all kind of equivalent to one another. And that's what it means that by the strength of the action potential never changes, but the frequency or how many we send in a period of time can change, and that does impact how our body responds and how we react. So, talking about the different phases of action potentials, we always start with some type of stimulus. The way that I like to think about this is a stimulus is some type of interaction of something that occurs, right? So, for example, if we go to touch the skin, we're almost touching, and we get super, 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 super close, but maybe there's like a tiny bit of touch, but not enough to actually feel it, right? So we're getting a little bit of stimulus, but until we press hard enough to actually start to feel it, we're not going to have any type of reaction. So that pressing hard enough to feel it, that stimulus needs to get to a point that's called threshold. Threshold is a specific point. It's set here because it's in relation to some of our um, gated ion channels that are going to drive this whole reaction. We'll talk about those in a second. But if we don't reach threshold, if we don't meet the requirements of the strength of the stimulus, we won't have an action potential. As soon as we meet threshold, though, some fun things are going to happen, and we're going to have an action potential, and it's going to get sent off down the neuron. If a stimulus reaches threshold, we go through a process that's called depolarization. Depolarization is a rapid change in the membrane potential where the inside of the neuron becomes more positive. So remember how we said, like right in here, negative 70, which cell type has a resting membrane potential of negative 70? That's going to be the neuron. And so if we have a stimulus in the neuron and we hit threshold, depolarization, this process is going to actually make the inside of the neuron go from being a negative number to a positive number. The way that it does that is by opening voltage gating, meaning the gate opens on these proteins based on the voltage. Voltage gated sodium channels open, which allow sodium to rush into the cell, causing the membrane potential to become less negative, moving all the way towards about positive 30, positive 40 millivolts. It hits a top point, it hits the peak at that highest point, the point of depolarization. Just like the voltage gated sodium channels had a spot where that caused them to open, they also have another set voltage that's going to cause them to close. So when it gets high enough, the sodium channels are going to inactivate and close. and voltage-gated potassium channels are going to open. Remember, we talked about potassium is in much higher concentration inside of the cell. So when those channels open, when potassium channels open, potassium is going to flood out of the cell. Potassium is positively charged, so as it floods out of the cell, we go back towards the negative, and that is called repolarization. So depolarization, we peak, and then we repolarize. So depolarization, sodium coming in, repolarization, potassium going out. The voltage-gated potassium channels, they tend to close a little bit slower as well. So we actually get a little bit of this overshooting where we go excessively negative. That's called hyperpolarization. And this plays a very important role in our action potential, which we'll talk about in just a second. Finally, after the hyperpolarization, we come back and we neutralize and we steady back into the resting state, back into the resting membrane potential. And that's getting back to the resting membrane potential. Staying there specifically is thanks to that sodium potassium ATPase pump, that one that we mentioned just in the last recording. Right there, sodium potassium pump actively moves 3 sodium out and 2 potassium in, reestablishing that resting state, that resting membrane potential. So why that hyperpolarization? The reason is refractory periods. So within our body, we don't want to be sending the same signal over and over and over and over again too quickly. If we send too many signals too quickly, they could kind of get gummed up and jammed and interfere with one another, and we wouldn't end up with nice, fluid, smooth messages going through the body. So we have these periods of time where either we absolutely cannot have an action potential occur, or it's more difficult for an action potential to occur. So the first one is the absolute refractory period. This is where during this time when one action potential is developing, we cannot under any circumstances develop another action potential during that time. There's no possible way because the sodium channels have been inactivated, right? After that period, during that hyperpolarization phase, we can have another action potential generated, but we need a stronger than normal stimulus. Think about it like this. If somebody lays a finger on your hand, right, you can feel it. If you sit there for a second, you start to feel it a little bit less and less unless your body adapts to it. Then all of a sudden they push harder and you feel it again. So as soon as they touch, that is a stimulus. It hits threshold. It causes an action potential. It sends a signal to the brain saying that something is touching us. And then while we're maintaining the same amount of pressure, that's like our absolute refractory period where we're not going to notice any change. The only way to get additional change to send more action potential is to press even harder and to get a bigger stimulus in order to overcome that hyperpolarization on the end side of it, and that is the relative part of the relative refractory period. The refractory periods also help with avoiding what's called back propagation, because we want these signals to always travel in one direction. So we don't want them traveling down both ends of the nerve. We want it just going in one simple direction. So this absolute refractory period and the inactivation of some of those ion channels helps to make sure that the signal travels in one proper direction. It also helps to avoid overstimulation of our tissue. So those were action potentials. We also have what are called graded potentials. These are small, localized changes in membrane potential that occur specifically in the dendrites or the cell body of a neuron. All right. So the dendrites, if we remember when we're looking at a neuron, the dendrites, all these little things sticking out here, those are the dendrites. They're the ones trying to receive signal from other sources to send it down. Graded potentials, these ones can add together. They can be either excitatory or inhibitory. So I mean they can encourage an action potential to be sent, or they can prevent an action potential from being sent. So we don't get a lot of questions specifically about graded potentials. More often than not, you're going to get some type of comparison between the two. So like this little chart right here. So graded potentials are the dendrites in the cell body. Action potentials are what go down the axon of the nerve. Graded potentials can be weak or they can be strong, but action potentials are all or nothing, always the same strength. Graded potentials decrease over distance, whereas the action potential stays consistent. Once we have an action potential, it stays consistent all the way down. Graded potentials can add up, action potentials cannot. Graded potentials do not require a threshold, action potentials do. Graded potentials are typically ligand or chemically gated or mechanically gated channels, whereas action potentials are voltage gated. Graded potentials, because they can sum together, they can add together, they do not have a refractory period, whereas the action potential has the absolute and the relative refractory period. Most likely things to get questions on is going to be the threshold as well as the signal strength, the variable or an all or nothing principle. Those are the two big ones that they like to ask questions about. Talking about the neuron itself, when we have the cell body, we have the dendrites, we have the cell body called the soma, and the part that actually travels to carry the signal is called the axon. Around the axon, we have these myelin sheaths. Myelin allows for something that's called saltatory conduction. Saltatory, in Latin, salta refers to jump. So literally, what we're talking about is the signal kind of jumping between these spaces of myelin. When we have a myelinated axon, because we don't have to depolarize and carry an action potential across the entire membrane, we just have to do parts because we can jump. It's much quicker when it's myelinated than when it's non-myelinated. That's going to make a connection back into spinal anatomy. When we talk about in spinal anatomy, we should have already talked about the different types of neurons, and how some of them are unmyelinated, like our pain fibers, our type C fibers, versus myelinated, which is a lot of our other sensory type fibers, as well as our efferent fibers are going towards the muscles, towards the other parts of the body. So the whole point of myelin is that allows that jumping, faster signal to get to where it needs to be. We can start to connect this with some of the pathology stuff. When we think about the myelin itself, what happens if the myelin becomes damaged or it starts to break down? Previously, messages that went really, really quick and got from sensation to destination super quickly are now no longer doing that if the myelin is being affected. And so certain conditions, like we look at MS is one of the most common ones, where the myelin in the central nervous system starts to break down. We end up with MS, we end up with lots of issues like decrease in muscle strength, decrease in nerve sensation, that kind of stuff, because we lose the myelin. So we lose the ability to quickly send those signals along the nerves. We'll finish up synaptic transmission in this one, yeah. So synaptic transmission, we have lots and lots of different types of neurotransmitters. This is mostly just for a little bit for memorization, knowing which neurotransmitters are going to be excitatory and which ones are going to be inhibitory. So the actual action or what happens, what kind of message gets passed along and what that message, what occurs, depends entirely on the neurotransmitter and the receptor. Mostly the specific type of receptor is what determines the outcome when we send a neurotransmitter. But kind of in general, acetylcholine, norepinephrine, epinephrine, glutamate, dopamine, and serotonin. These are all of are primarily our excitatory neurotransmitters. So all of these are going to have an excitatory pass another message along. They're going to excite the next neuron, the next nerve, the next cell to pass the message along. Compared to GABA and glycine, which these are our inhibitory neurons. These are the ones where if they're being released, they're actually going to stop and prevent signals from going through. A super common medication that most people have heard of is gabapentin. Gabapentin is primarily used for nerve type pain, and that's because gabapentin is an artificial GABA, which that GABA is an inhibitor, so it inhibits the neurons and inhibits signals, specifically pain signals, from going to the brain. So that's a good way to remember some of our inhibitory neurons. Within the peripheral nervous system, we're going to come back and talk about a lot of these. Like the neuromuscular junction, we'll talk about. Neuromuscular junction uses acetylcholine. The autonomic nervous system uses acetylcholine and norepinephrine primarily. Different types of receptors. These are the names that you'll see. Muscarinic receptors. What we want to know about the names of the receptors is what system they're associated with and also what neurotransmitter they're associated with. So muscarinic receptor means that it is a receptor that accepts acetylcholine and it is part of the parasympathetic target organ, are the muscarinic receptors. Nicotinic receptors, these are acetylcholine receptors, but they are for skeletal muscle fibers and preganglionic, sympathetic, and parasympathetic neurons. Norepinephrine and epinephrine are also known as adrenergic receptors, and we will see those either as alpha receptors or beta receptors. Beta receptors primarily refers to epinephrine. Alpha more with norepinephrine, but that's not a hard rule. So just we want to connect. If you see adrenergic, it's referring to norepinephrine or epinephrine. Alpha, beta, they're all just essentially AKAs of one another. So that's what we want to remember, just for understanding the word sake of things. Different types of synapses. We'll come back to a lot of these, and we'll see these specifically. A lot of this is kind of introductory. We have electrical synapses, such as gap junctions. Those are found primarily in the heart. Chemical, which cause release of neurotransmitters. We'll talk about those with skeletal muscle. Calcium-dependent exocytosis. This is where calcium binds to vesicles. Vesicles are little holding containers for neurotransmitters, and then they cause those vesicles to leave this cell. We'll see that as well when we talk about the process of skeletal muscle contraction. Excitatory and inhibitory postsynaptic potentials. Biggest thing we want to hear is just word recognition. Knowing excitatory postsynaptic potentials refers to a specific event that brings the neuron closer to threshold, making it more likely to fire an action potential. Inhibitory postsynaptic potential is a hyperpolarizing event, moves the neuron further from the threshold, making it less likely to fire an action potential. So we want to remember excitatory So an EPSP makes it more likely to fire an action potential. An IPSP makes it less likely to fire an action potential. And that's about as far as we'll go with all of that. Okay, we will jump into motor function and continue on in the next one.